Hi, my name is Netanya Keil, and I'm a fourth-year PhD student in the McIntyre Lab at the University of Florida. And today I'll be presenting SQANTI-reads, a tool for evaluating the quality of replicated long-read RNA-seq studies. So SQANTI-reads leverages SQANTI-3QC to evaluate the quality of replicated long-read RNA-seq experiments. And today I'll focus on presenting 4 features of SQANTI-reads and how we apply them to real data sets. So feature 1, SQANTI-reads groups mapped reads based on their internal junction patterns into groups that we call unique junction chains or UJCs. The number of reads assigned to each UJC is recorded, and these UJCs can be tracked across samples and conditions. We also classify them as novel or known splicing patterns relative to a provided reference transcriptome. Feature 2. SQANTI-reads also identifies putative unannotated transcripts that are not present in the reference annotation. So a novel UJC that is represented by a large number of reads indicates a novel splicing pattern that the user might want to follow up on. So SQANTI-reads uses a customizable logic tree to fish out these high-confidence novel UJCs from the data. Feature 3. SQANTI-reads quantifies variation around annotated junctions by calculating the distance of each read donor and acceptor from its nearest annotated junction. We then calculate the mean standard deviation and coefficient of variation around each annotated donor and acceptor and classify it as a clean or messy junction based on the calculated coefficient of variation. And feature 4, SQANTI-reads produces several multi-sample summary files and visualizations that can be used to evaluate the quality of the data set. And these visualizations can be faceted by experimental factors like technology to evaluate different aspects of the experimental design. And SQANTI-reads also performs a principal component analysis using the quality metrics to identify which metrics drive sample variability. And so, as I mentioned before, reads that share all internal junctions are grouped into a representative unique junction chain, and the number of reads associated with that UJC is recorded. So here I'm showing an example of a set of reads mapped to a gene, which is shown in gray, and the corresponding annotated transcript in that gene, which is shown in black. And so these reads are then assigned one of the SQANTI-3 structural categories. So we have full splice match, or FSM, which means all junctions in the read match an annotated transcript. Incomplete splice match, or ISM, which means that there are junctions missing on either the 5' or 3' end, or both. Novel in catalog, or NIC, which represents a novel combination of known donors and acceptors. And novel not in catalog, which means that a read has at least one novel donor or acceptor. And so we then group these reads into unique junction chains. And so in this example gene that I'm showing, there are 104 reads and 5 unique junction chains. So in general, we see that for a high quality data set with a well annotated reference transcriptome, about 70% or more of the reads represent an annotated splicing pattern, or FSM. And in this experiment, we see that this is consistent across the 3 replicates. And this is the WTC11 benchmark data set from lrGAS. However, when we collapse the reads to UJCs, we see that only a small proportion of the UJCs have annotated splicing patterns, and the majority of UJCs actually show novel splicing patterns. This is because, in general, we see that there are a small set of full splice match UJCs represented by a large proportion of the reads, and there are many novel UJCs that are represented by few reads, and in many cases, we see that these novel UJCs are represented by only one read. So if we go back to our example, we could see that there's an FSM that represents about 40% of the reads in the gene, and an NNC that represents only 2% of the reads in the gene, which matches the pattern that we usually see. However, there's another group of UJCs of interest, which are the novel UJCs that are represented by many reads. And so to identify these novel UJCs of interest, we use a data-driven approach with a customizable logic tree to find them. So first, we select genes with greater than 100 reads, Then we select all of the novel UJCs in these genes and identify those UJCs which represent more than 20% of the reads in the gene and have more than 80% of the maximum number of junctions observed in the gene. And the reason that we have this threshold for the number of junctions observed in the UJC is because we care about, we don't want to capture fragments that might be fragments of a longer transcript. And so all of the thresholds that I mentioned in this logic tree are customizable, and the user can adjust them to be more or less selective if they would like to. And so we apply this logic tree to the WTC11 iPSC cell line benchmark data, and we found 424 putative novel transcripts, which are highlighted on the scatter plot. On this scatter plot, each dot represents a novel UJC. And so on the y-axis, we have the percentage of the maximum junctions in the gene. And on the x-axis, we have the percentage of total gene coverage. So about 3/4 of these putative novel transcripts that we identified are classified as novel in catalog, which means that the majority of these putative novel transcripts that we find are actually just a novel combination of known splice sites. And then the remaining quarter of the putative novel transcripts that we identified are novel not in catalog, which means they have at least one new splice site. And finally, or close to finally, to quantify variation around splice junctions, we use a metric known as the coefficient of variation, where we calculate the mean and standard deviation of the distance of read donors and acceptors to its nearest reference. And so based on the calculated CV, we then classify reference donors and acceptors into 3 categories. So the first one is reference match, where reference match, where all the reads match the annotated donor or acceptor. Then we have CV equals 0, where all of the read donors and acceptors match each other, but don't necessarily map to the annotated position. And finally, CV greater than 0, where all of the read donors and where there's some degree of variation around the annotated donor acceptor, and a larger CV corresponds to a greater degree of variation around the annotated donor acceptor. So both the reference match and CV equals 0 we consider to be a clean splice site, and CV greater than 0 we classify as messy. And so it is known that splicing is perturbed during disease, and particularly in cancer cells and during oncogenesis. So we hypothesize that there is increased messy splicing in cancer cells. And so in a preliminary analysis, we use a publicly available long-read RNA-seq dataset of colorectal cancer tumor samples and adjacent normal tissue. And we calculated the CV of the annotated donors and acceptors. We then put all of our CV metrics into a principal component analysis. And on the PCA plot, you can see that the cancer samples separate from the healthy tissue samples. In the interest of time, I'm not showing the data, but we do see that there's increased messy splicing in the cancer samples. And this is an ongoing work, and we're trying to identify which genes and which specific donors and acceptors are the messy ones in the cancer samples. And so in conclusion, SQANTI-reads, I hope, is a useful tool for the community to compare different facets of replicated long-read RNA-seq experiments. And we present new methods for quantifying junction variation and identifying putative unannotated transcripts. And I'd like to thank Dr. McIntyre and the McIntyre Lab, as well as the Canessa Lab, Dr. Canessa and Carolina Monso, for helping me integrate this work into the SQANTI-3 universe. I'd also like to thank my funding sources, and SQANTI-reads is open source and available for download on this SQANTI-3 GitHub. I can take questions now.
Okay, we have time again for one very short question, if there is one. Yeah.
What's the data set size, and how fast does SQANTI-reads run it? It sounds like it already assumes you're giving it input aligned BAM file. So basically runtime and memory.
Yeah, so we have different modes in which you can run SQANTI. So you can start from a fast queue, and it will do and you can put your preferred aligner, and it will do the alignment for you. But the way that we suggest, the fast mode that we suggest that users use is actually to parallelize running SQANTI-3 first on your samples, and then using the output of SQANTI-3 to produce the summary files and the summary visualizations. And I don't remember exactly the runtime, but I think for like 5 samples with 2 million reads each, it took about 5 to 10 minutes to run.
Let's thank the speaker again.